This is your Chapter 6 lecture video on neurotransmitters and their receptors. This chapter follows Chapter 5, which introduced you to the different types of signaling that can occur at the end of an axon, so the different types of synaptic transmission. We talked quite a bit about chemical neurotransmission and the different steps that happen in order to package a neurotransmitter that's coming from the cell body and carry it down to the synapse. We also talked about the importance of depolarizing the membrane in order to have calcium rush in so that the vesicles would then fuse with the membrane. And so it's important to keep this in mind that in order to have chemical neurotransmission, that these are the things that are happening. But then what I introduced to you in the last chapter was that the actual neurotransmitter can either be synthesized at the cell body and packaged into vesicles, or it can be added into the vesicle right at the synapse. So it just depends. And so this is the chapter where we're going to introduce you to the different types of neurotransmitters that we often find in nervous systems. And the book focuses on predominantly vertebrate organisms. So we're going to be focusing on neurotransmitters that you would see primarily in humans. There are, I hope you keep in mind that there are over 100 different types of neurotransmitters that we know of, and there are still some that can be discovered over time or different permutations of them. When we do discover a new neurotransmitter, we place them into 3 different categories. So amino acids, amines, and neuropeptides. Why is it important to know the neurotransmitter and the receptor type of your favorite neuron? Mostly because you want to know how that neuron is going to function. And so if you are interested in studying neurological disease or mental health disorders, those are basically iterations of the nervous system that are not functioning properly. And in order for the nervous system to function, as we talked about last time, much of that function relies on chemical signaling in the form of these neurotransmitters. So that's why it becomes extremely important to really understand the neurotransmitter at the chemical level and also to understand the receptor to some extent. So what is a neurotransmitter, or what defines a neurotransmitter? So they actually, in order for a molecule or chemical to be defined as a neurotransmitter, it has to meet certain criteria. And so the criteria that must be met are outlined here, but there's a few more that are outlined in the note section of the slide. So be sure you take a look at the note section so that you can get the full picture. But starting with the first one, pretty simple, right? If it's a neurotransmitter, it has to be present in the presynaptic neuron. So when you're doing your first pieces of research on a new neural system, a new neuron partner, presynaptic and postsynaptic partner, you would want to stain the presynaptic neuron using antibodies to determine what type of neurotransmitters are found at that particular synapse. You would also, in order for it to be classified as a neurotransmitter, you would also want to know that when there is a depolarization and calcium is coming into the cell, that the neurotransmitter that you're studying is actually released. So in order to be a neurotransmitter, it has to be a calcium-dependent release. In addition to the neurotransmitter being present at the presynaptic neuron, you also have to have at the synapse the enzymes that are responsible for the synthesis of the neuron. So Those are three things already. So having the neurotransmitter present, having the enzymes for the transmitter present, and also that when you stimulate the neuron, that we have a depolarization event and calcium influx that causes the release of the neurotransmitter. The other portion of the story is, of course, the postsynaptic side. And so, in order for a chemical to be defined as a neurotransmitter, it has to have a very specific receptor located on the postsynaptic side. And more importantly, when the neurotransmitter binds to those receptors, that the response of the postsynaptic cell is some type of postsynaptic potential, like we talked about, either excitatory or inhibitory, like we talked about in chapter 5. The other ways that we define a neurotransmitter is experimentally. So we can define a neurotransmitter by placing the actual transmitter or an agonist of the transmitter into the dish where we have our neuron and having the postsynaptic response, right? So having the release of that neurotransmitter affect the postsynaptic potentials. The other thing that we do to determine, yes, indeed, this is a neurotransmitter, is to block the release of the neurotransmitter altogether. So using an antagonist that will block the neurotransmitter. The end result of that, if it is truly a neurotransmitter, is that the postsynaptic cell will not respond. So you will have either no postsynaptic response or you will have reduced the postsynaptic response depending on how much you block the release of the neurotransmitter. And so these neurotransmitters don't, you'll find they have multiple jobs in the cell. They don't have to just be a neurotransmitter. So we have some neurotransmitters that function as other things for the cell. For example, glutamate and glycine and ATP. They can all function as neurotransmitters, but the glutamate and glycine, of course, are both amino acids, and adenosine triphosphate, I think you know quite well, has other roles in the cell. So before we get into your chapter, I actually included a few slides at the beginning to help you understand and kind of put everything together. The different receptors, of course, that we're going to be talking about in this chapter. I also gave you slides to help you understand how somebody would even find neurotransmitters within a nervous system. So what kind of experimental techniques would we use to determine a new neurotransmitter or the neurotransmitter of your favorite presynaptic and postsynaptic pair? Just as a reminder, we are talking about the neurotransmitters that will bind to these postsynaptic receptors, and that those receptors can be either ionotropic or metabotropic. And so go back to chapter 5 if you're not quite remembering what those two things are. What you're going to see, just like with the ion channels that we talked about for action potential, you're going to note that these receptor types are very similar. So the ionotropic receptors are very similar in structure, and the metabotropic receptors are very similar in structure to themselves. So here we are talking about a multi-subunit channel that forms a pore for an ion. As far as the kinetics of the channel, these ionotropic channels can be very fast acting, so about 1 to 5 milliseconds to have passage of charge. The ionotropic receptors are not going to activate a signaling pathway in the cell, so that's what this is all about. So it's not going to do that. And that if the ionotropic receptors are bound by the neurotransmitters, they tend to open, they rarely close, and that We typically find these ionotropic receptors or these type of receptors on the postsynaptic side, and we tend to call them a ligand-gated channel, right? Because you have to have the neurotransmitter bind to it. As far as how many molecules does it take to open the channel, usually one molecule of the neurotransmitter is enough to open the channel. So think back to the vesicular release that we just talked about in chapter 5, and remember that each vesicle can have up to 1,000 molecules of neurotransmitter within them. So we can possibly activate very strongly neurotransmitters when we have ionotropic receptors. In the metabotropic case, we have these are usually protein receptors that are called 7-pass transmembrane receptors. So they basically have protein subunits that pass through the membrane 7 times. They are G protein coupled receptors. And so we're going to be talking in chapter 7 about what is a G protein, what does it do, and I've introduced it to you in the last chapter, but I haven't told you all the things that it does. So metabotropic receptor binds to the neurotransmitter. It takes a varying length of time for this receptor to respond, right? Look at the difference here. We've got a 1 to 5 millisecond response for ionotropic receptors. For metabotropic, we said it was slow. It's 100 milliseconds in some cases to days. It takes days for some of these receptors to actually open. There is always some type of signaling pathway that's involved. And then, the effect of these particular receptors being activated could either open or close ion channels within the cell. They are also postsynaptic, but they can be affected by voltage. They can be resting channels, so they can have different roles, even though these are metabotropic type receptors. They can have an effect on these types of channels, right? So ion channels, voltage-gated channels, and resting channels. The last piece was in that list was looking at how many molecules does it take to open the channel. So one neurotransmitter will open many channels. So just to kind of follow up with that, we are going to get into this later, but I do give you this now, this chart now, so that you can have it. In chapter 7, we're going to actually introduce these signaling pathways that we commonly see in metabotropic receptors. So there's 1, 2, 3 that we're going to talk about, but for now, just know that this is coming in chapter 7. I would maybe put a note that we will come back to it so that you can have the actual figures that go along with these signaling pathways. I think it'll make it a lot easier. Okay, so let's put it all together. What do these neurotransmitter systems actually look like? How are they studied? How does one figure out which is the primary neurotransmitter that's being utilized by a neuron pair? And so that's what the next few slides is all about, right? Figuring out the neurotransmitter structure as well as the receptor structure. So you're looking at a couple of different types of diagrams. The neurotransmitters and the receptors are both either, in the case of a neurotransmitter, they can be an amino acid, but they're proteins. So proteins take on very different shapes and structures depending on the individual amino acids that make up the protein. So you can see here this beautiful crystallography diagram of a receptor. And this is actually a glutamate receptor. And then you can see that in a different conformation here. And then if we were to look more in the I guess, ball and stick drawing of it, or not ball and stick, but actually cartoon drawing of the receptor, you would see that it would pass through the membrane multiple times and then, most likely curve so that you could form an ion channel. So there's a lot of information here, but my question was more, how do we figure all this out? Okay, so there are different methods in the laboratory that can be used to study any kind of protein, but especially a neurotransmitter or the neurotransmitter receptor. And that is the very first thing is just to find where is it located, right? Which neurons contain this neurotransmitter. So you can use immunohistochemistry, which we've talked about before. And the idea here is that you are taking antibodies to determine where in the cell is this particular protein located. So location, location, location. Where is the neurotransmitter located? Is it located at the axon hillock, at the axon? Is it already at the terminal? And so immunohistochemistry really helps quite a bit to help you find either the receptor or the neurotransmitter. Remember that you have to use antibodies that are specific for these two different types of proteins, but then once you have labeled them, you should be able to see them. The other technique that's often used is not at the protein level, which was what we were just talking about, but at the RNA level. So this is a pretty powerful tool because when you use it, you can basically answer the question, when is the gene for this neurotransmitter being actively transcribed and translated? When is it actually being used? So this is in situ hybridization. And in situ hybridization uses a complementary RNA strand, which you can see here. And that complementary RNA strand is going to bind to the strand that's found in the cell for your neurotransmitter or for your receptor. Once it does that, you can either attach a radio label tag to it, which is what you see here, or a fluorescent tag. And so you basically can see under the microscope, which is what you see on the bottom panel, the lighting up of whatever gene product that you are looking for. So this is messenger RNA. These are what the in situ hybridization images typically look like. It's easy to get these confused with the fluorescent images that we would see for immunohistochemistry. You would really need to be told by the paper that you're reading if this is mRNA staining or if this is protein staining. And so if they tell you protein, you know that this is an immunohistochemistry. And if they tell you, nope, it's RNA, then you know it's an in situ hybridization. So basically, you can take any nervous system. As you can see here, there's a few different nervous system slices for you to look at. And you can bind to them the antibodies that you are most interested in studying. And then you can see where the actual color deposit is left behind, and that is where that mRNA is being expressed. So it's a pretty powerful tool because you can literally see where a gene is expressing. And so here, this is a cross section of the embryo, and you can see that there's already some activation of a gene, a specific gene in different regions of the embryo. So continuing on with how do we study neurotransmitter systems, one of the ways that, after you've determined where those neurotransmitters are located, which is what we would use the in situ hybridization and what we would use immunohistochemistry for, the next thing is to really figure out how is that neurotransmitter affecting the physiology of the connection, right? So how does it make it function? Is it an excitatory response, or does it evoke an inhibitory response? And so we really have quite a few ways that we can approach studying the effect of the neurotransmitter on the postsynaptic cell. We can study the timing of the release of the neurotransmitter and how it affects the postsynaptic cell. We can study the amount of that neurotransmitter that is released and how it would affect the postsynaptic cell. And so we can do it a couple of main, I guess, groupings. The way we actually approach these questions would be through in vitro studies or in vivo studies. So in vitro would mean studying the neuronal connections outside of the body. So actually studying them in culture dishes, or to study in the animal itself. So I've talked a lot about having a neuron in a dish or having a brain slice in a dish. And so you can do that. We can actually keep a brain tissue and neuronal tissue alive in a petri dish when it's bathed in the saline solution that you would normally have those cells exposed to. And so we can keep them alive for some time and we can record from them, right? So we can do some electrophysiology on brain slices. And so you can very easily stimulate the synapse and measure how much of the response on the postsynaptic response that you have. So what is the amplitude that you have on the postsynaptic neuron? You can also put onto this slice different drugs, so antagonists or agonists of a neurotransmitter that you think might be functioning in this location, and then record and see what happens. Do you still get a response or do you not? You can also collect from tissue, as we've already talked about, but you can actually take brain tissue and neuronal tissue and you can extract protein and mRNA from that tissue and determine whether or not the neurotransmitter you think is made in this area is actually present. And so we'll talk about that a little bit in a moment. And again, you can also do immunohistochemistry here so that you can see if the neurotransmitter protein you think is present is actually there. So this would be an example of in vitro study of timing and amount of release, where we would record from a specific neuron in a dish or a brain slice in a dish after we stimulate to see how it affects the postsynaptic response. We also have in vivo tools that we can utilize. And so these in vivo, so you're actually recording brain function within the animal. And so there are some limitations here. We obviously, it would be difficult to do this with humans, but this has been done primarily with primates. At least some parts of this has been done with primates, mice, and rats. Fish as well, so one example would be where we can actually place an electrode into different brain regions using a cannula and then record from those different regions, either directly recording after a stimulus, so recording how the postsynaptic response is responding, either excitatory or inhibitory, or we can use the same setup, but we could drip onto the slice or through the cannula, and again, a drug, an agonist or an antagonist, so that we can show a specific neurotransmitter is functioning in that particular area, right? So that's, again, very invasive. And so we would, you typically see something like this with mice or rats and occasionally primates, although to be honest, in current times where the National Institutes of Health is moving away from using primates as a model system. We can also study timing and release of neurotransmitter in different brain regions using positron emission tomography or PET scanning or functional MRI. You can at least see which regions of the brain in functional MRI would actually be responsive under certain stimulus. But with positron emission tomography, you're basically radio labeling a ligand that's administered via IV to the patient, and then if neurotransmitter is released during the PET scan, the radio labeled ligand will decrease in um, in receptors because of displacement by the endogenous neurotransmitter release. So it's kind of a backwards way of thinking of it, right? So you have a radio label um, agonist, and so when you stimulate and the neurotransmitter that is normally released is present, then the radio label will not be able to bind. So you wouldn't have as much immunolabeling of the PETs of that region on the PET scan. So again, looking at how much neurotransmitter is released, where is it released, how long is it released in that area. Lots of different questions that you can ask with these both in vivo and in vitro studies. Another in vivo, well, not in vivo, but in vitro study, so outside of the body, very interesting one that I don't think your book actually mentions. It's a very new technology that it involves, I don't have a picture for you, but it involves the development of what would be a small developing brain tissue in culture from stem cells. So there are techniques that are now available that are used in developmental biology labs where you can actually take human pluripotent stem cells and under very, very stringent conditions where those stem cells are receiving signaling molecules that would force the stem cell into down the road of developing a brain tissue. You get in culture these what we call organoids, and they're actually, in this case, because you're talking about using signaling molecules that are important for development of a brain, you get cerebral organoids. And so there's a little bit of controversy right now with these organoids, although they've been extremely useful for more recent questions. I mean, you essentially have a baby brain in a dish, and you can really, do all of these types of questionings that we're talking about here, but using human tissue, or at least cells that were taken from a human to create these baby brains. And so using organoids was essential in understanding how Zika virus affects the development of a human brain. And so lots of different different types of drugs that are utilized in the brain were tested with these organoid cultures. And so there's definitely some use of the organoid model system, just like the brain slice. But there have been some questions about the ethics of having essentially a human brain that you're growing in a dish. And there are some limitations, of course, with the fact that it's not a fully functional human brain. It's just the beginnings of what would be the brain. Okay, so there was a lot on that slide, but basically I keep using these terms agonist and antagonist. So this is my one slide to make sure that you know that a lot of our understanding of which neurotransmitters were used or are used in specific brain regions or different regions of the body come from these pharmacological studies, right? So every neurotransmitter has some other pharmaceutic, either naturally occurring or not, that can mimic the activity of that neurotransmitter, or the opposite's true, where there are naturally occurring or pharmaceutically produced antagonists of the neurotransmitter. And these drugs are essentially, they're very important for our understanding of how the neurotransmitter functions at any given synapse. So, I mean, the first question they help us answer is, well, is this particular neurotransmitter the one that's used primarily by this axon? Um, so that's like question number one. Question number two, three, or four could be, well, how is this neurotransmitter used? Is it used alone, or are there other neurotransmitters that are also functional at this synapse? So an agonist is a drug, I guess to define them, they are drugs that facilitate the effect of the neurotransmitter in this case. They mimic, we say, that the typical ligand that the receptor will bind to. And then the antagonist would be the drug that would do the opposite. It would inhibit the effect of the neurotransmitter. And so you can have a competitive inhibitor, you can have a non-competitive inhibitor. And so there's also that to consider when you're talking about antagonizing or blocking the effect of the neurotransmitter using an inhibitor. They can act in a competitive way where they bind to the receptor, the same receptor that the neurotransmitter would bind to, or they can bind non-competitively by binding at some other site, either on the receptor or somewhere else along the axon, and somehow affecting the postsynaptic membrane so that the neurotransmitter is not capable of binding to the receptor. Either way, whether you use agonists or whether you use antagonists, what you're asking is first of all, you're trying to identify what neurotransmitter is present at this particular synapse. And so, understanding that, then I guess the other side to that is that if you know which neurotransmitter is being utilized, the next question then is, well, if my favorite synapse involves, for example, acetylcholine, well, then which of the acetylcholine receptors are functional? So a lot of the work in this chapter involved the pharmacology of determining the neurotransmitter, but also determining which receptors are activated or inactivated by that neurotransmitter. So what is the receptor partner for the neurotransmitter? And so it's very common that the neurotransmitters will bind to more than one type of receptor. And so, for example, here you have acetylcholine. Acetylcholine can actually bind to a nicotinic acetylcholine receptor, and they can also bind to a muscarinic acetylcholine receptor. How do we figure that out? Well, we used pharmacology. And so curare is a poison that was actually utilized by many indigenous populations that utilize poison darts. And at the tip of the dart, you can place curare, and that will negatively inhibit these receptors. So whenever the individual is hit by one of these darts, the curare will bind to the receptor and negatively, they will prevent acetylcholine from actually binding to the receptor. So it's considered an antagonist of acetylcholine. And so because of the fact that curare is doing this, then it was pretty easy to figure out that the skeletal muscle contraction, for example, utilizes a nicotinic acetylcholine receptor and therefore also binds to acetylcholine. In the case of muscarinic receptors, they actually can be positively affected by muscarine, so they can be antagonized by muscarine, or they can be antagonized by atropine. And so atropine is actually belladonna. It's from a plant. From a belladonna plant, and it will antagonize the muscarinic receptors. And so we find muscarinic receptors, for example, in your cardiac muscle tissue. And so again, using these two different pharmacological agents, one is an antagonist and the other is as an agonist, we were able to determine long ago that the heart muscle or cardiac muscle is utilizing an acetylcholine receptor, muscarinic receptor, in order to function. The other examples that we'll get to later in this chapter are glutamate. Glutamate actually has 3 different receptors. So when you are studying a specific neural connection in your organism and you know that glutamate is present, the next question is, well, which of these receptors is actually working at that synapse? And so again, utilizing these pharmacological agents, we're capable of helping us determine what receptor and what neurotransmitter signaling partner is working at any given synapse. And so here's an example from a paper that I published with a colleague last year. It says 2019. I think they actually released it in 2020. But in my worm, we were trying to understand whether or not glutamate was functional at a specific synapse in the worm. My colleague had done some electrophysiology with a specific neuronal connection within the worm. And what he found was that when you bathe a antagonist of AMPA, the AMPA receptor for glutamate. So again, glutamate has 3 different receptors, NMDA, AMPA, and kainite receptor. So these are the 3 here. And so when you bathe with the antagonist of one of them, this AMPA one, you get no action potential. And so you can see here's the normal condition where you get an action potential. And once you put that antagonist on, the action potential's gone. And so it essentially indicates that the AMPA receptor may be one that is utilized by this particular synapse, because you don't get the same response when you use the antagonist for the other type of receptor, the NMDA receptor. And so this is just an example that I'm showing that we had a glutamate synapse. So we did the immunohistochemistry, which you see here, and the glutamate is in red. So we knew that these synapses were glutamatergic synapses, but we didn't know which receptor was functional at that particular synapse. So there's a lot more that can be done. I think in the 1970s, thereabouts, with the I guess explosion of molecular techniques that really started when we were able to do PCR, polymerase chain reaction. We basically were capable of studying the nervous system then at the genetic or molecular level. And so we knew for a long time which were the active neurotransmitters and neurotransmitter receptors at many different synapses, but we didn't really understand, the actual structure of those receptors and structure of the neurotransmitters. And so this is where a lot of the molecular techniques that you've learned about in your freshman biology, or if you took 3202, which is another biology course, these are the types of things that you would learn in the cell molecular research labs that we have. Things like protein extraction, gel electrophoresis, Western blotting, all of that is protein characterization. You would learn RNA extraction, cDNA, complementary DNA synthesis, PCR, which is polymerase chain reaction of a specific gene sequence. And then once you have that gene sequence, you can do in situ hybridization like we talked about. You can knock out the gene. A lot of talk has been happening around CRISPR-Cas9, so you can use that to study the receptor or the neurotransmitter. So the sky is the limit here in regard to what we can actually do to study neurotransmitters and their receptors. Okay, so that was sort of the introduction to how you can study these neurotransmitter systems in the nervous system. Just a little bit of theory. So before we get into the chemistry, Sir Henry Dale had been one of the leaders at the beginning of our studies for neurotransmission, and he proposed a hypothesis that was called later the Dale's principle, that that for every neuron there was only one neurotransmitter. And so at the time that Dale proposed this principle or theory, there really wasn't a whole bunch known about the neurotransmitters. So to be honest with you, when he proposed this theory or principle, there were only two that we knew of, and that was acetylcholine and epinephrine. So there was still quite a few more to be discovered. And so, thinking back to that first slide of, well, what makes a neurotransmitter, his idea was that if you look at any one neuronal system, the signaling neuron, right, and the receiving neuron, that there is only one, and that there's one for every neuron. Well, we know now that that's not necessarily true. We would say, I think it would be better to say that neurons that are released from neurotransmitters that are released from a neuron are going to be released at all the axonal connections for that neuron. So let's say you have neuron X, and you know that it releases acetylcholine. It's going to release acetylcholine from any connection that it makes, okay? And that may be the main neurotransmitter that it utilizes. But we know that there are lots of co-transmitters that are also released with the major neurotransmitters. Sometimes, you can have two different neurotransmitters at a synapse, but we don't now, in this day and age, with all the techniques that we just talked about, we know now that Dale's principle is not necessarily true. So again, he was very instrumental in purifying and studying acetylcholine, and so this was his idea that that this particular synapse functioned because it had an acetylcholine connection, right? Acetylcholine receptors that were responding to the release of acetylcholine, and that was the only thing it would respond to. And so now we know that, again, that there are multiple neurotransmitters at the synapse, and that for the most part, that it's still a good rule of thumb. So we say that his principle is still valid in current neuroscience studies, because when we're looking at diseases within the nervous system, especially diseases that are very complex and involve different regions, let's say, of the brain, it's important for us to focus one at a time, what might be going on. So like one neurotransmitter at a time, one neuronal connection at a time, one region at a time. So it's a good rule of thumb to start off with, looking for a specific neurotransmitter that may not be functioning properly, right? So we can think of dopamine and Parkinson's, which is what Dr. Martinez will talk about when he comes for a visit. We can think of quite a few other dysfunctional, problems within the nervous system, like the myasthenia gravis, or different myasthenic syndromes that have some, problem with acetylcholine either release or acetylcholine reception. So yes, it's a good rule of thumb, but it's not necessarily it's not 100% true. So here's some other examples that I have. Okay, so in general, I would restate, this is Dr. Acosta's statement, that Dale's principle is, in general, all axonal branches of a neuron release the same neurotransmitter substance or substances, but I wouldn't say that would be the only one they release. All right, so that was a long introduction to the remainder of the chapter. And so what I would recommend is, so that you don't get overload, that you stop after every neurotransmitter and make sure that you have completed that portion of your homework. So on your homework for the problem set, there's a space where you can answer, what the neurotransmitter is made from. So, what is the base compound that it's made from? What is the enzyme that is utilized to produce the neurotransmitter? And which receptors does it bind to? So, what is the actual name of the receptors? And then, in addition to that, I think it's the last part of it, is what type of receptor is it? Is that a metabotropic receptor or an ionotropic receptor? So it's my way of getting you to help condense these notes a little bit. And so just before we get into the specific biochemical divisions of these neurotransmitters, you should know that there are basically, these primary, what we consider primary neurotransmitters. There's 3 different types, and so there's the classic neurotransmitters, many of which are small amino acids, right? There are some monoamines that are in this sort of classic. These are the ones that we tend to focus on because they are often used within the human nervous system. But then there are also small peptides, and so you can see there's quite a few here. Oxytocin is one of my current most interesting ones that I'll try to bring some more recent research into our discussions with oxytocin. So there's the classic neurotransmitters that you learn about, but also other molecules, like insulin also, that function as neurotransmitters. And then there's also gases, nitric oxide and carbon dioxide, that can function as neurotransmitters. And yes, there are co-transmitters, as we just talked about. And so you'll occasionally see the release of the main neurotransmitter along with, another secondary transmitter. Okay, so the main biochemical types, so that was sort of dividing them into groups. We can also divide them up by their chemistry. And so your book introduces them to you that way. And so there are basically 3 major classes of neurotransmitters. And I love this chart because it gives them all to you very organized. And so you have what I would call the amino acids. And so you have glutamine, aspartate, GABA, glycine. You have also in this category, you would have ATP, which is a purine. It's not an amino acid. But you'd also have acetylcholine, which is a small neurotransmitter. So these all are grouped together under the amino acids, even though, only one of them is actually an amino acid. Then you have these amines. So you have these biogenic amines, and they are either, so let me stop for a moment. They are monoamines, but we can subcategorize them into catecholamines, indolamines, and imidazolamines. Okay, and so you have dopamine, norepinephrine, and epinephrine as your catecholamines, and then you have serotonin, and you have histamine. Right? Acetylcholine can be considered in this particular group, but we tend to treat acetylcholine separate from them, and just by looking at the structure of acetylcholine in comparison to these more traditional amines, you can see the difference between them, and then we'll talk about a little bit what the difference in their chemistry. So amino acids group, amine group, and then this last group there's not very many of them, but there are small peptides that serve as neurotransmitters. So these are, of course, they're small peptides, but they're larger than these amines and amino acids that are utilized. And so we'll talk a little bit about some of those. There's also a discussion that of these types with the neuropeptides. So that last group, there's so many of them, but if you take a look at them, they can vary in the length, they can vary in the number of amino acids. So they're just all very different. So we'll probably introduce only a couple of these peptide neurotransmitters. But if you take a look at them, you've probably talked about them in some of your other courses. So I know there's been reference to the endocrine course, And so I think that these are lots of neurotransmitters that you would see in the endocrine system. So I wanted to put this table here at the beginning of our series of slides that we have for each of these neurotransmitters. I think this is a nice organizing table, so it would be one that's probably going to help you quite a bit with your homework assignment. But you can see the neurotransmitters that we're going to be talking about. You can see what type of effect does it have on the postsynaptic cell. So is it an excitatory postsynaptic response or inhibitory? You can also see what are the precursor molecules that are utilized to synthesize the neurotransmitters, and then what the rate limiting step for that synthesis is, which I'll just say right now, most of them are going to be limited by the presence of the enzyme. So you can see most of these are enzymes that are responsible for the production of these neurotransmitters. And so without the presence of a fully functioning enzyme, that's going to be your rate limiting step for production of the neurotransmitter. There's also here, how do we remove the neurotransmitter from the system? So what are the inhibitors of them, or the molecules that are responsible for breaking them down? So which enzymes are utilized for that? Or if there are transporters that move them out of the system. And also what their vesicle looks like. This part I wouldn't worry about so much. I'm not going to ask you questions about whether or not it's a small vesicle or a large, dense vesicle. So these are more important to me, that you know how they're made, basically what is the starting material, what is the enzyme, and then how do they get removed from the system. Okay, so we're going to start with acetylcholine. It is, as we've already talked about, it is the best studied neurotransmitter. But what I'll tell you is that it's best understood at the neuromuscular junction. So this last category here, where you have the cranial nerves and motor neurons that are going to be synapsing on the skeletal or smooth muscles, or you have these spinal motor neurons that are going to be coming from the spinal column that will again synapse at the neuromuscular junction on the skeletal muscles. So these are probably the most well-known acetylcholine receptor or acetylcholine synapses. What we don't know very well is the function of acetylcholine within the CNS, within the brain neural connections. So there are some regions that we have some research that's been done to try to understand the acetylcholinergic synapses within the CNS. And so we know, for example, that there are synapses within the ganglia that are in the visceral motor system and within quite a few other sites in the brain. But again, many of them we don't fully understand. So in the septum, there are neuronal connections within the hippocampus that utilize acetylcholine as their neurotransmitter. And then there's the nucleus basalis that has projections that are in the isocortex. So these axonal projections in the hippocampus are going to synapse on postsynaptic neurons that are important for memory function. We know, for example, with some of the studies that have been done, that in Alzheimer's patients where there's pretty far progression of the disease, you have these acetylcholine synapses that are severely affected in those brain regions of the hippocampus. We also think there are some papers that suggest that these acetylcholine synapses within the septum or within the hippocampus may enhance the coding of a memory. And so there's some papers that have shown that as well. The isocortex also is thought to be involved in memory function. And so again, there's just a lot that we still need to figure out, I guess, in terms of what exactly is going on within the CNS connections. And so just to give you an idea, I like to give you, the structure to go along with the actual description of where those connections might be. And so this is the basal forebrain. So here's that nucleus basalis, and these would be where the neurons would originate, and then they would send projections outward into the different regions of the cortex. And so you have the nucleus basalis, you have the medial septal nucleus, and nucleus of the diagonal band. You also have the hippocampus, and you have projections that will come there to the hippocampus, and then you have the cerebellum and other areas within the brain that these are actually a part of the brain stem. So in the pons, where there are a collection of neurons in the upper pons that are important for arousal, attention, learning, and reward. And then some of these projections are involved in voluntary limb movement. So we have, again, those neuromuscular components here. And then these particular connections from the basal forebrain are going to be involved, they're going to have axons that terminate in the hippocampus, which you can see there as we talked about, that have some role, as we've already suggested, in learning and memory, but also in sleep. And then the nucleus basalis will send connections out to the cortex that will modulate arousal. They will modulate sensation of, I guess, aversive stimuli. And these are interesting, these nucleus basalis projections, because there are papers that suggest that they're particularly vulnerable in age-related diseases. So these were the ones that I was talking about, that they may be very severely damaged in Alzheimer's and Parkinson's patients. But yeah, I'm not sure that we understand fully what exactly they're doing there. So to talk about the chemistry, so how is acetylcholine synthesized, and then how is acetylcholine broken down? Your book actually gives you this diagram, and I think it does a very good job explaining how acetylcholine is actually synthesized. So it's synthesized from acetyl-CoA, and it involves the use of choline acetyltransferase, or CAT is what you'll hear people talk about it as CAT. So it's abbreviated. If you find, for example, with all of these neurotransmitters, if you find the enzyme within this synaptic terminus, then most likely this is a acetylcholine or cholinergic synapse. And so you can see acetyl-CoA and choline will be utilized together along with choline acetyltransferase to produce acetylcholine. And the acetylcholine, then, this is all happening at the synaptic terminus, that will be transported into the vesicles using a acetylcholine transport channel. And so the vesicles, this is an example of a neurotransmitter that's packaged at the synapse. You'll notice, though, just some people I know are taking cell biology. You'll notice that where do we get acetyl-CoA from? We actually utilize acetyl-CoA during cellular metabolism. And so the metabolism of sugars is also important for neuronal function. This is just one example where acetyl-CoA will be utilized from metabolic processes. And then choline will get recycled. And so you notice here that there are these choline transporters in the membrane of the presynaptic neuron that are transported. So this is a co-transporter where you have choline coming in, utilizing sodium as the driving force to bring in choline. what what is happening, right, is that when acetylcholine is released at the synapse, then the acetylcholine will bind to its receptors in the postsynaptic membrane, and we'll talk about those in a minute. But acetylcholine esterase is responsible for the breakdown of acetylcholine at the synapse. It's thought that acetylcholine esterase can cleave about 5,000 molecules of acetylcholine per second. And so it's basically cleaved back into acetate and choline, and then choline will then be recycled back into the neuron so that you can continue to make acetylcholine. So this is an essential process where it's constant production, but then use and recycling of the choline. So the CAT, choline acetyltransferase, and acetylcholine esterase are very tightly controlled enzymes that are very important for the function of acetylcholine at this particular synapse. So the acetylcholine chemistry is quite interesting. It brings up a little bit of history, and so with acetylcholine esterase, there are different nerve gases that have been utilized throughout warfare history, and so there's sarin and soman, which are both irreversible acetylcholine esterase, so the enzyme that's responsible for breaking down. They are irreversible acetylcholine esterase inhibitors. So essentially, they're designed to disperse as a vapor or a cloud in the spray form. And so that allows entry into the body via skin contact or inhalation. And so essentially what's going to happen, if they are inhibiting acetylcholinesterase, you're going to have buildup of acetylcholine and overstimulation of the cholinergic synapses all throughout the CNS and the PNS. And so often that can be fatal. And so if an individual is able to survive from one of these types of attacks, they tend to have chronic fatigue, diminished heart function, and memory. It also has effects on memory, as we've talked about. It's interesting along these lines, and so, we at least in this particular area, the idea is to try to understand what types of biological warfare agents are available for use. And so there are still some cases where we've seen the use of nerve gases or some type of neurological compound that will interfere with function of the nervous system. And so there was a pretty recent, I think this was March of 2018, situation where there was a former Russian spy that was living in England at the time with his daughter, and they became gravely ill and were sent to the hospital. It was unclear at first what was going on and how, why they were sick or why they were ill. And so after investigation, they figured out that they had been exposed slowly over time to a nerve agent that also inhibited acetylcholine esterase. It's Novichok, which is, I believe, Russian for newcomer, newbie, or novice. And so Novichok was an interesting story. It's something out of the movies, I guess, for some of us to think about. It was placed on, I think, the door handle of their apartment. And so they had frequent contact with the Novichok agent, and it was slowly causing the increase of acetylcholine within their systems. So, how do they work? At least, how does acetylcholine um, bind? What type of receptors does it have to bind to? So, it can bind to the nicotinic acetylcholine receptor or the muscarinic acetylcholine receptor. And so, I do want you to know a little bit more about these two receptors since we have the information for them. Nicotinic acetylcholine receptors are ionotropic receptors. So they are going to form a pore that allows for passage of ions once all the different subunits of the receptor come together. So you can see that the nicotinic acetylcholine receptor is made up of 5 units, so 5 subunits, 1, 2, 3, 4, 5, and that the alpha subunit, these red subunits, actually have the acetylcholine binding sites. So, how does this work? So they're going to come together in the membrane. They have membrane spanning regions, each of them. They actually pass through the membrane, although it's hard to see here. There's 1, 2, 3, 4 different membrane spanning regions, and they will form a pore that is in a conformation that is closed until acetylcholine binds. So when acetylcholine binds, these portions of the channel will twist and open, and that will allow for passage of charge. How do we study acetylcholine receptors? And actually, how do we determine whether or not we have an acetylcholine or cholinergic synapse? You can use the agonist nicotine. So nicotine actually will bind to these binding sites for acetylcholine, and they will activate these postsynaptic sites by opening up the receptor. The antagonist is curare, and curare will actually block the receptor or block the binding sites so that you cannot get acetylcholine to bind to it. And so this type of ionotropic receptor and the biochemistry of it, why is it important? Well, many of the ionotropic receptors have this type of structure, and so acetylcholine or nicotinic acetylcholine receptors actually have helped quite a bit for our understanding of how an ionotropic receptor looks, what it looks like. You also have muscarinic acetylcholine receptors. These are metabotropic receptors, and so we find these in places where you have smooth muscle primarily, and so they are metabotropic. So again, they're going to be bound to these G proteins, and then they're going to result in some internal cascade of proteins that are going to get activated, right? There was that domino effect when you have a metabotropic receptor. Here, the structure looks very different. We're not creating a pore. You have a bunch of alpha helices that are passing through the plasma membrane multiple times. So this is actually, it's hard to tell here, but it's passing through the membrane 7 times. And then there's a binding site for acetylcholine. This particular binding site will also bind to muscarine. Muscarine is an agonist and will allow for the receptor to function as it would when acetylcholine is bound. Or there are antagonists, so atropine serves as an antagonist for these smooth muscle muscarinic acetylcholine receptors. And so, some important pharmacology that is useful for us on a relatively, I guess, common, I don't want to say everyday basis, but common drug use that interacts with these receptors. Atropine is actually as we talk about here, it's an antagonist of this receptor, and so, or of the acetylcholine in this case, and so it will block the muscarinic acetylcholine receptor. And this is utilized during pupil dilation, and so when you've gone to the eye doctor, you get a little bit of atropine to dilate those pupils. Scopolamine is useful for motion sickness. I tend to actually have really bad motion sickness when I'm in a bus. And so scopolamine will work directly at the synapse. And so in the areas of the brain which would control nausea and sensation of nausea that causes vomiting, you actually would have at those particular synapses these metabotropic receptors, and scopolamine will block these receptors so that the acetylcholine is no longer capable of binding, and therefore you lose the sensation of having motion sickness. There's also eprotropium. Okay, that is utilized for treatment of asthma. And so the idea here is that, again, the eprotropium will bind to the receptor in the muscles that are around the airway, and that'll cause relaxation of those muscles so that you can increase the flow of air through the airways. And so we talked about other uses of pharmacological agents that interact with the acetylcholine receptor pathways. And so, one of them, again, Clostridium botulinum, being utilized in the cosmetic industry to reduce the number of wrinkles that you have in the facial region or wherever they inject the Botox. And so essentially this is functioning at the acetylcholine synapse. But remember that this is not a matter of binding to the receptor or interacting with the acetylcholine directly. So the botulinum toxins will actually interfere with SNARE complex function. And so you're not able to get fusion of the vesicles to even release the neurotransmitter. And so because of that, it reduces the amount of contraction in that particular area because you're not releasing the neurotransmitter. And then we also, I don't know that we talked about cobra venom, but we talked about other types of toxins. And so alpha bungaroo toxin is capable of binding competitively, and so it is an antagonist of the nicotinic acetylcholine receptor. So this is the neuromuscular junction, and so it would irreversibly bind and essentially cause overstimulation, would not allow for stimulation, and therefore you'd have you would essentially paralyze the individual with the alpha bungaroo toxin that is in the cobra venom. We also introduced myasthenia gravis and its effect at this particular synapse. And so with there were quite a few myasthenic diseases we talked about in chapter 5. And so this one, myasthenia gravis, was the autoimmune dysfunction where you have antibodies that are produced that block the nicotinic acetylcholine receptor binding sites. And so acetylcholine is no longer able to bind. And so that results in the symptoms that you can see up here a little bit with this particular patient. So there's muscle weakness, particularly in the eyelids, the limbs, and with respiration. Myasthenia gravis can be treated with organophosphates. And it's also interesting. So we talked about how the acetylcholine synapses within the CNS have been problematic in Alzheimer's patients. And so there is some push to use organophosphates with those patients to help with the deterioration of the acetylcholine synapse as a part of their treatment. There's also a use of acetylcholine esterase inhibitor. And so if you can keep acetylcholine in the synapse a little longer, then it's possible that maybe you could have increased binding of the acetylcholine to any of the receptors that are not affected by the antibodies. And so it really would just depend on whether or not, the number of acetylcholine receptor antibodies that were produced. The use of acetylcholine esterase inhibitors and organophosphates is a common treatment for other places where we also see the need for binding of acetylcholine a little bit longer. And so here's just from this particular chapter, again, going along with, we've already introduced myasthenia gravis, so just showing you how the actual function of the synapse is affected. Here's normal postsynaptic responses for an individual that does not have myasthenia gravis. But when you look at the postsynaptic response in individuals that do have it, the action potential peaks are much more reduced. They're not as consistent with each activation. And so you get reduced activation over time. And then if you treat with the organophosphate or if you treat with an acetylcholine esterase inhibitor, you are able to rescue a little bit of the postsynaptic response. And so we know that myasthenia gravis, this pathology that you see here, is the result of a presynaptic problem and not of a postsynaptic problem, because if you look at the number of mini end plate potentials in a person who has myasthenia gravis versus someone who does not, the actual number of vesicles that are released is the same. And so, or the amplitude of voltage is the same. So this is not a problem with acetylcholine release. As we already know, it's a problem with the receptor being inhibited. Okay, so that was our story with acetylcholine. Again, we know quite a bit, but there's also some places there where there's still lots that we need to learn, particularly within the cholinergic synapses that are within the brain that have implications for learning and memory, and then also arousal. So we'll move now into the biogenic amines, and we'll start off with the catecholaminergic neurons, or the catecholaminergic systems. The biogenic amines, what you're going to find is that these are neurotransmitters that are going to regulate a wide variety of brain functions, and also they can also be active in the peripheral nervous system. Because they are going to be utilized in so many different areas where brain function is going to be affecting mood and attention. These are neurotransmitters that are often implicated with the psychiatric disorder, and because of that, there's quite a bit of pharmacological interest in these biosynthetic pathways, and also interest in understanding the receptors that are utilized by these different cholinergic or catecholinergic neurotransmitters. And so these pathways also, we see that these are pathways that are impinged upon during drug abuse, or are involved with drug abuse. And so they are interesting. They provide lots of interesting study questions, I guess I should say, that relate to human brain function, but particularly in regard to behavioral disorders. In the case of catecholaminergic neurons, these are all neurotransmitters that have a common structure, and so they are made from a precursor, which is tyrosine. The three neurotransmitters have as a part of their structure a catechol group, which is essentially a benzene ring. So you have that here that has two hydroxyls, so two hydroxyls and two side chains that contain amino groups. And so you find that with each of these, so with dopamine, norepinephrine, and epinephrine, this is pretty standard, this catechol that's a part of their structure. So that's why they fall into this category of catecholaminergic neurons. Epinephrine, you know, probably as adrenaline, and then norepinephrine as noradrenaline. We base their basic synthesis is pretty straightforward. You have to have tyrosine available in order to produce any one of these within the catecholaminergic neuron. And so, what is the rate limiting step? Honestly, the rate limiting step is having tyrosine and having tyrosine hydroxylase so that you can produce DOPA. Once you have DOPA, you can pretty easily move forward. You do have to have other enzymes, and so I think really, I guess the beginning of this is though having that tyrosine available. And how are you going to get tyrosine? Well, that's going to be more diet related and through phenylalanine hydroxylation. So phenylalanine, again, being an essential amino acid. And so phenylalanine will be increased in brain using phenylalanine hydroxylase. And so phenylalanine hydroxylase essentially is the enzyme that converts phenylalanine into tyrosine. And so just some interesting information here as well that relates to the clinic. And so when you have a reduced amount of phenylalanine hydroxylase, that leads to phenylketonuria, which is treated dietarily by adding phenylalanine to the diet. So again, very important to have tyrosine to produce these different catecholaminergic neurotransmitters. And so I would like you to know this pathway. And so it's not important to me that you actually draw the structure. I do want you to know that the actual neurotransmitters themselves, dopamine, norepinephrine, and epinephrine, they all have this structure that we call a catechol. But basically knowing that the precursor is tyrosine and that tyrosine hydroxylase will convert tyrosine to DOPA, which is dihydroxyphenylalanine. Dihydroxyphenylalanine will decarboxylate, and so we're going to remove carbon dioxide using DOPA decarboxylase to produce dopamine. So there's your first catecholaminergic neuron. Dr. Martinez is going to be talking to us more about dopamine and dopaminergic neurons that are known to be dysfunctional over time with Parkinson's patients. Dopamine then can be utilized as the precursor for norepinephrine. So dopamine beta hydroxylase will produce norepinephrine, which is noradrenaline, right? And then phenylethylamine N-methyltransferase is utilized to produce epinephrine from norepinephrine. Okay, so just to give you some, I guess, more slides to use for studying, I've put the pathway here for the production of each of these neurotransmitters so that you have it for studying, right? So each of them depending upon tyrosine to be present. What I also want to talk about is before we get into the indolamine serotonergic neurons, I want to also talk about the fact that norepinephrine will be functional more in the brain, and epinephrine would be more functional in the periphery. And that for all of these biogenic amines for the most part, breaking down, right? So we were talking about enzymes that break down these neurotransmitters, breaking down the bulk of these biogenic amines is going to be done using monoamine oxidase, or MAO for short, monoamine oxidase. And so these will deaminate the catecholamine or catecholaminergic neurotransmitters. So, as a part of these biogenic amines, we're also going to introduce the endolamines and the serotonergic neurotransmitters. And so, I'm introducing the pathways to you, and then we'll talk about the different neural connections that utilize all of these neurotransmitters in the next couple of slides. So, with the serotonergic neurotransmitters, serotonergic or serotonin is produced utilizing tryptophan as its base component. And so tryptophan then would have to be available within the axonal terminus. And so tryptophan hydroxylase becomes important in this case. So here we have again a hydroxylase when you compare how these biogenic amines are produced. So tryptophan will then be converted into 5-hydroxytryptophan, and 5-hydroxytryptophan will be decarboxylated. So again, very similar here, using an aromatic L-amino acid decarboxylase to produce 5-hydroxytryptamine, which is serotonin. So serotonin, we actually will abbreviate a lot in the papers as 5-HT. And so this tryptophan is taken up by neurons using transporters, and then that then will be converted into the 5-hydroxytryptophan using the tryptophan hydroxylase. So all of the neurons that utilize serotonin would have tryptophan hydroxylase in them. We find serotonin is utilized throughout the nervous system, but we particularly find it within the enteric nervous system of the gastrointestinal tract and the central nervous system, particularly with the raphe nuclei. So serotonin is going to regulate intestinal movement, but also in the CNS. So that's what it's going to do at the GI tract. In the CNS, we're going to find that serotonin will affect mood, it will affect appetite, sleep, and cognition. And, we talked about what's going to break down these catecholamines. We talked about monoamine oxidases, but with serotonin, we're going to be talking about serotonergic inhibitors. So let's look at dopamine. So again, we're going to look at each of these one at a time. Dopamine will have axonal, I should say cell bodies that are originating in the ventral tegmental area that will send projections to the nucleus accumbens, the hippocampus, the frontal cortex, the amygdala. We also have neurons within the substantia nigra that will extend axons to the caudate nucleus, the putamen, globus pallidus, and the motor cortex. So if we think about what all of these different regions have been implicated as far as, what types of function do they control within the nervous system, the nucleus accumbens is an area we know that is important for pleasure, and so this is a region where we know that this particular neuronal connection is important when looking at addiction. And so we know, for example, heroin and cocaine will bind in these particular dopaminergic synapses, or will bind within these regions and then affect addiction, and so you get an increase in patients who are addicted to heroin and cocaine. You get an increase in the amount of tyrosine hydroxylase, and therefore you get an increase of dopa within the system that underlies the addictive nature or the addiction, uh, the pleasure that is activated in those particular regions. The amygdala is sort of the seat of avoidance and fear conditioning, and so again, dopamine being utilized in this region to um, help stimulate this feeling of fear, but also avoidance as well in relation to stimuli that provide fear. We also have the substantia nigra. These regions, the caudate nucleus, the putamen, the globus pallidus, and the motor cortex, all of these are regions that we know are important in regard to motor function. And so Parkinson's patients tend to show a severe decline of dopaminergic neurons in these regions of the substantia nigra. And so we understand dopamine quite a bit within its function of motor control. We also know that the ventral tegmental area will send projections to the tegmentum. So that's not, I didn't list that here, but it will send projections to the tegmentum, which is basically a region that is going to contain a group of neurons that's close to the midline of the midbrain. And this is again areas where you have reward circuitry. So again, going along the lines that we were talking about earlier with addiction. So reward circuitry, cognition, motivation, pleasure, addiction, intense emotion, love. These are regions, the tegmentum, the connections from the ventral tegmental area do provide with dopamine as the underlying cause, intense emotional responses of love particularly. And these are regions that we know are disrupted in patients or not functioning properly in patients who have schizophrenia, different types of mood disorders. So schizophrenia, and also things like attention deficit disorders, so motivation, right? Parkinson's are also, we've already talked about those as well. We know the substantia nigra, so again, not on here, but the substantia nigra, in addition to these projections that affect motor function, that they also will affect eye movement, planning of movement, so motor planning, reward seeking, learning, and then addiction. So dopaminergic neurons are involved in quite a few things, but I think if we had to categorize them, I would place them into, involvement in reward, pleasure, motivation, mood disorder, and neuromuscular or movement, motor control. The types of receptors that are utilized by these dopaminergic pathways are all metabotropic. So these are all dopamine receptors. So they're D1 or D2 family. You don't have to worry about these smaller D1 and D5. You don't have to worry about that. But D1 and D2, and that they are all metabotropic receptors. And so here you can see a lot of those projections that we were just talking about, and where, the place where they're coming from, the substantia nigra and the ventral tegmental area, and that they're projecting out into all those different regions that we just talked about. And then knowing again that these are mostly connections within the prefrontal cortex, and then also some that are in the brainstem. Okay, so yeah, so I always put this in here. So yeah, if dopamine or dopaminergic neurons are mediating the sensation of love and intense emotion, then the question I often ask at this point in class is, if we're in love, was it really love, or was it just dopamine that was maybe overreactive that day? So that's me. That's my silly joke. Okay. We also then will move into norepinephrine. So noradrenaline or norepinephrine, which primarily we will see the use of norepinephrine as a neurotransmitter in the locus coeruleus. So this is going to be found in the pons. And just some background for norepinephrine, remembering that this is also a hormone. It's a hormone and a neurotransmitter. And so, it's as you probably have learned in other classes, it's going to ready the body for action, right? So it's going to be decreased during sleep, but it's going to be increased during stressful or dangerous times. So flight or fight, right? So, thinking about our current situation and the amount of stress that we're under, we probably have quite a bit of activation of our noradrenaline, norepinephrine, or epinephrine neurotransmission centers. And so, how is it functioning? We know that in the brain, with these particular connections to the thalamus, the hippocampus, the amygdala, the striatum, the cerebellum, the spinal cord, that we are going to be looking at norepinephrine affecting increase of arousal and alertness, enhancing formation of long-term memory, so thinking, right, hippocampus, and focusing attention, increased restlessness and anxiety in your body, right? So the connections that would synapse in different body regions, you're going to get increase in heart rate, blood pressure. So that's what's going to be happening. We already talked about the synthesis of norepinephrine, and that it is a part of the pathway that requires tyrosine. And so I don't think I need to go over that again. This particular neurotransmitter, again, is going to be broken down by the monoamine oxidases. And so I think that's it. Oh, the receptors for norepinephrine are also they're specific. They are metabotropic. They are beta receptor and alpha receptors. And there are very specific or interesting pharmacological agents that interact with these two types of receptors. So beta adrenergic receptor or alpha adrenergic receptor. Beta blockers are utilized for people that have glaucoma or migraines or cardiovascular problems because of the presence of norepinephrine in these particular regions, and because the outcome of the use of norepinephrine is increasing, for example, heart rate and blood pressure. So beta blockers are often used to combat those symptoms. Alpha adrenergic blockers are used for cardiovascular disease and also psychiatric disorders. So they're often used as a sedative to sort of bring you back down and sedate the individual. So this is what that looks like. So I basically, for all of these, give you the actual location of the cell body, and then where the axons project to, and then synapse. So, and then this is the diagram of what that looks like. So you have the locus coeruleus here, and you can see the cell body sending out axonal projections in these different areas that we talked about. Okay. Oh, so I guess I had... Oh, okay. So then epinephrine, so that was norepinephrine. And so now with epinephrine, we see that there's not as far-reaching, activity of epinephrine, as comparing it to norepinephrine. So if we take a look at epinephrine, we have that it's found at lower concentration within the brain, particularly in comparison to all the other neurotransmitters we've talked about so far. It is synthesized from norepinephrine, which is what we just talked about. And so you'll have phenylethanolamine and methyltransferase that produces epinephrine. Its function is again with fight or flight, and that you do get increased heart output and pupil dilation with epinephrine. And so you're going to also have regulation of respiration and cardiac function with epinephrine as well. And so what are the overall behavioral outcomes of an epinephrine neuronal connection? This is going to affect emotional responses, particularly fear, anxiety, stress, enhancing of long-term memory. And so, again, not as far-reaching as all the other neurotransmitters. You can see that with the projections, which are coming from the medulla. And so there are some that will project outward to the spinal cord, and then a few that mostly project to the thalamus, the hypothalamus, and the pons. So the receptors, sorry, I forgot to tell you, the receptors for epinephrine are alpha and beta adrenergic receptors as well. So they are metabotropic, and they are found in the sympathetic nervous system. So the connections, right, the actual receptors for the epinephrine are found within the sympathetic nervous system, and they're part of the autonomic nervous system's, flight or fight or freeze and respond behaviors. So let's take a look at what some of these receptors look like. We've been talking about some of them being metabotropic, or for the catecholamines, for them being metabotropic. And so you can see here, this is the dopamine metabotropic receptor. So very similar to what we talked about with acetylcholine, where you have these alpha helical regions that are spanning the membrane. These are 7-pass membrane receptors. So there are 1, 2, 3, 4, 5, 6, and probably another one that we can't see. 7 that form the receptor and then have a binding pocket, in this case for dopamine. This is the D3 receptor, and this particular receptor will bind to dopamine, but then it's also known, so the receptor itself, to be functional during hyperactive states and during repetitive behavior type, movements. For other catecholamine neurotransmitters that we talked about, norepinephrine and epinephrine, these are images of the beta adrenergic receptors. And so you have the membrane spanning regions again during the inactive state, and then during the active state, where you have these compounds that will come up to the receptor itself and interact with the receptor. So then looking at the indolamine serotonin, we've already talked about its biosynthesis, but I put the biosynthesis here again for you. Remember that the tryptophan is going to be taken up by the neurons via a transporter, and so you're going to have tryptophan in the axonal terminus that will be hydroxylated to 5-hydroxytryptophan, and then that will be decarboxylated to serotonin, or 5-HT. Remembering that these neurons are found in the raphe nuclei of the pons and of the upper brainstem, and that they're going to project axons to the forebrain. These particular connections are going to function in sleep and wakefulness. There's also going to be axonal projections that will impinge upon areas that are important for mood, particularly affecting depression, the mood disorders like depression and anxiety. There's also, they do not, serotonergic neurons do not utilize monoamine oxidase to break down serotonin. So you have selective serotonin reuptake inhibitors that will break down serotonin. And so these are antidepressants. And so they essentially leave within, if you inhibit the reuptake of serotonin, then you basically allow for the serotonin to remain at the synapse and therefore increase mood when you are in a depressed state. So, for example, Prozac is an example of a selective serotonin reuptake inhibitor. And so these are the raphe nuclei is where the cell body would be for the neuron, the neuronal connections that have serotonin as its main neurotransmitter, but then they project axons out to the thalamus, hippocampus, frontal cortex, amygdala, septum, striatum, and cerebellum. And so, again, serotonin being utilized for sleep, wakefulness, also mood disorders. So we know serotonin, and actually I've studied it more often with its function in circadian rhythm. So that's the sleep wakefulness. In motor behavior, emotional states or mood disorders, arousal also. We have some indication that serotonin will function for mental arousal. And then it also mediates satiety. So you get a decreased amount of food consumption when you increase serotonin. We know that serotonin, therefore, can be used to treat different types of eating disorders. And we also know that the receptors for serotonin can also be bound by LSD, causing hallucination. So again, with its role in sort of mood or state of being. And then the receptor types for serotonin, they are a large family. So there's about 7 different families of receptors. These are just serotonin receptors. You don't have to know all these different numbers. They are all metabotropic except for one. There's one receptor that is actually ionotropic. So what do they look like? So you actually have an image from your book. And so here is an example of the metabotropic receptor. And so, as I said, LSD can actually bind to these receptors, at least the metabotropic receptor, and then that will lead, again, trigger hallucination. The ionotropic receptor looks very similar to the one that we talked about with acetylcholine. So you have the 1, 2, 3, 4, 5 subunits that come together to form a pore. And so for the ionotropic receptor, your book talks about how this is a ion channel, which is what ionotropic receptors are, but it's not selective for any specific ion. The serotonin ionotropic receptor is not selective for a specific ion. We know that Zofran is utilized to prevent postoperative nausea. I have had to use it before after surgery, and also it's utilized in chemotherapy patients because nausea is a side effect of chemotherapy. And so basically Zofran works through this ionotropic receptor that in the serotonergic neurons. And so this is what the pathways of the serotonergic pathways look like. And again, I think we've seen this one already. I may have repeated this twice. So you have the cell bodies that are coming from the Raphe nuclei going all over the cortex, but also the thalamus and the corpus callosum, and then you have also projections into the brainstem and the pons. You'll have cell bodies within the pons that then project to the spinal cord. Okay, so this last group of neurotransmitters are the transmitters that we're going to be talking about with our paper. And so I wanted to go ahead. I know this is a very long recording. Again, highly recommend doing this recording in pieces. And so glutamate and GABA are our last big neurotransmitters. These are a part of the neurotransmitter group that are amino acids. And so we're talking about them together because one of them is the most common excitatory amino acid, which that's glutamate. And then the other is the most common inhibitory amino acid that we see in the neurotransmitter and then in the nervous system. And so they often work together in order to mediate behavior. And so one of the things you're going to read about in this next paper, which the first group is going to be presenting this. I think that what you'll notice is that these two neurotransmitters work to basically modulate each other. And so I think it'll be an interesting paper to read that will showcase learning and memory and how some of the receptors that we use for the glutamatergic systems are useful during learning and memory. How are they utilized to mediate learning and memory? Glutamate is a non-essential amino acid. It does not cross the blood-brain barrier. And so glutamate is going to be synthesized at the axonal terminal. And we're going to actually synthesize it using glutamine metabolism using an enzyme called glutaminase. And so glutaminase is going to be extremely important for the production of glutamate. Again, it's the major excitatory neurotransmitter in the brain, and it works by binding 3 different types of receptors. And so it just depends on which neuronal connection that you're studying. It does not bind to all of them at the same time necessarily, but one neuronal connection might use a certain receptor, and then another will use a different one. So the different receptors are the AMPA or NMDA channels, and then the kainate receptors. The AMPA and NMDA channels are sodium, calcium, are permeable to both sodium and calcium, and they result in depolarization of the membrane because of the influx of those positive charges. GABA is a major inhibitory neurotransmitter in the brain, and so GABA is GABA aminobutyric acid, and so we usually just call it GABA, but it is able to activate ion channels that are permeable to chloride ions. And so again, you get this balance of excitatory and inhibitory signaling within the brain. And so we'll be focusing on the use of these 2 different types of neurotransmitters with our learning and memory paper. So if we take a look at the different types of receptors, so the glutamate neurons are found all over the brain, as the slide indicates, but there are different types of receptors that it will use. And so I mentioned the 3 ionotropic receptors, AMPA, NMDA, and kainate. But then there's also metabotropic glutamate receptors, so both ionotropic and metabotropic. And so the AMPA receptors, the NMDA receptors have a very characteristic structure, and so the receptors themselves have a magnesium plug that will be moved whenever the membrane potential becomes depolarized. And so you can see here that, one of the steps, a function of this receptor is that when you have a depolarization event, in order for the channel to actually become useful, the magnesium plug has to be removed. And so you have to reach a certain threshold in order for that to happen. Once the magnesium plug is moved, then you can have passage of ions through the channel. All of that's going to, of course, necessitate glutamate binding to it, and so there's a little bit more to these glutamate receptors, at least with NMDA, than meets the eye. So these are some images that your book provides for you, a little bit more detail than the cartoons that we had on the last slide. And so you can see that NMDA is a calcium channel that is blocked by magnesium, and that with increase in sodium in the cell, so again, depolarizing, right, the cell is depolarizing, then you get removal of the magnesium plug, and then you are allowing calcium to influx into the cell from external environment, and that will then cause an action potential. And so there are NMDA receptors that we find throughout the cell that function in this way, and so it is slightly different than the other ionotropic receptors that we talked about, because it has this dependence on the membrane potential unplugging the magnesium plug. These NMDA receptors, what we're going to see is that as you increase neuronal activity or activation of these receptors, you can effectively encourage the production, so transcription translation of more NMDA receptors that would be placed into the membrane. And so in the paper we're going to read, that's going to be one of the underlying mechanisms for the establishment of a memory. And so that's going to be, I don't want to give too much away, but we're going to be looking at insertion of more NMDA receptors, because the more you activate or stimulate, the more receptors that would be expressed in those neurons. These NMDA channels, again, will allow for calcium or sodium to pass through, but we just talked about calcium, or your book introduces you to it as a calcium channel. The NMDA receptor can be affected by psychoactive drugs like PCP or dextromorphan. So again, playing a role in addiction. But then also, I guess, just structure-wise, there's 3 different subunits present in the NMDA receptor, the NR1, NR2, and NR3 subunits that come together to form the channel itself. So there are, as we said, both ionotropic, so we talked about the AMPA receptor, the NMDA receptor, the kainate receptor. There's also metabotropic glutamate receptors. And so these, whether we're talking about ionotropic or metabotropic glutamate receptors, the response of the cell is still excitatory, and so you're going to elicit a excitatory postsynaptic potential. So again, looking at this metabotropic receptor, not too different from the ones that we've looked at already. You do have this really interesting adaptation, and I actually don't know the story here. I don't know why this particular metabotropic receptor evolved to have this, we call it the venous flytrap domain, where you have opening and closure states that will allow for binding of glutamate. So here's the open state, and then when glutamate binds, it sort of closes off as a venous flytrap would, and that also changes the spacing here between the membrane spanning regions, and allows for activation of those proteins inside of the cell that I've been telling you are like a domino effect. And so a little different structure, but for the most part, you have alpha helices that are spanning the membrane region. So it's a multipass membrane receptor that activates proteins within the cytoplasm. It just has this cool Venus flytrap domain. So again, we're going to be coming back to glutamate when we talk about our next paper, but looking at synthesis of glutamate within the cell, we've already talked about how glutamate does not cross the blood-brain barrier, and so it has to be synthesized at the terminus. And the way this is going to happen is essentially by utilizing astrocytes, remember those astrocytes, that will take up glutamate from the synapse, and glutamine synthetase, okay, so in the astrocyte, will convert glutamate to glutamine, and then glutamine will be transported out of the astrocyte and taken up by a transporter on the glutamatergic neuron. So we're basically recycling, and we're bringing in glutamine. So glutamine will then be converted to glutamate using glutaminase, and glutaminase will will produce glutamate, and glutamate will then be transported using these glutamate transporters in the vesicle. So you'll get the glutamate packaged into the vesicles ready to dock and to fuse again if needed. And so this is pretty straightforward, but it involves the astrocytes that we talked about before that are going to be instrumental in breaking down the glutamate that was released to begin with. This is what the AMPA receptor looks like. It is a sodium channel, and so these AMPA receptors also excite, resulting in an excitatory postsynaptic potential. So they activate, they excite the postsynaptic neurons. Again, we're going to be talking about this receptor along with the NMDA receptor in the paper that we're reading next. You can see that this is an ion channel, and so you are going to create a pore. This is a much smaller ionotropic receptor, but the pore size is the size that you would need for a sodium ion. And then the binding of glutamate to the actual receptor itself is a little bit, it has what we call a clamshell, very similar to what we just looked at for the metabotropic receptors, but you have glutamate binding to this region up above, and that will change the conformation of the protein so that you open up this small pore that will allow for sodium to pass through. And so again, with these with the amino acid neurotransmitters, we're talking about glutamate. We'll also talk about GABA, so gamma aminobutyric acid, aka GABA, which is the major inhibitory neurotransmitter that is found within the brain. GABA will activate these chloride ion channels, and so binding to the receptor will open up the channel and allow for chloride to move in, therefore hyperpolarizing the membrane and resulting in an inhibitory postsynaptic potential. There are binding sites on these GABA receptors for things like benzodiazepine compounds, so Valium and Librium, barbiturate binding sites that are utilized to reduce the number of seizures. The pharmacological agents that reduce the number of seizures that happen. So these are the ionotropic GABA receptors, but there are also metabotropic receptors for GABA, which are potassium channels, not chloride channels. Again, we'll be talking about GABA when we look at the next paper. this is the figure that's found in your book, so you can see that whenever we stimulate the presynaptic neuron, you do actually get an inhibitory response, and then uh, you can have the activity return after that synapse has completed its binding of GABA to the receptor, but binding of GABA to the receptor results in the influx of a negative ion, and that will reduce the ability for you to elicit another action potential at that synapse until the GABA has cleared out. And so you can see again the pore that's formed. You have 1, 2, 3, 4, 5 subunits again. So again, looking very much like the ion channels that we've seen already. So if we take a look at the biosynthesis of GABA, we essentially start with cell metabolism. And so the byproducts of glucose from the tricarboxylic acid cycle or the citric acid cycle will result in the production of glutamate. Glutamate then will then be converted to utilizing the enzyme glutamic acid decarboxylase. And so you have glutamic acid decarboxylase in any of the neurons that will be utilizing GABA as one of its major neurotransmitters. And so GABA then will be transported into the vesicles at the axon terminus and then released at the synapse. As far as removal of GABA from the synapse, just like we talked about with glutamate, there are transporters within the astrocytes that will utilize sodium to bring in glutamate, and also there are transporters that are available at the neuronal membrane as well, so not just the astrocytes. So just some other important differences. The production of GABA here requires the glutamic acid decarboxylase, but glutamic acid decarboxylase is not fully functional unless you have pyridoxal phosphate present. And so pyridoxal phosphate is going to be absolutely essential for the function of that enzyme to produce GABA. Pyridoxal phosphate is derived from vitamin B6. So if there's a deficiency in vitamin B6, that can lead to diminished GABA synthesis. This is important because it is pretty important for infant development. And so there were quite a few infant deaths that were linked to the omission of vitamin B6 from infant formulas. So the absence of vitamin B6 greatly reduced GABA content in the brain of these developing infants, and that caused seizures that ultimately were fatal in many of the cases. And so once GABA is synthesized, it can then be transported into the vesicles, as we talked about, and utilized at the synapse. But this is incredibly important during development, as we talked about at the beginning of this, that both glutamate and GABA are going to function together within the nervous system to sort of modulate function. And so giving you examples that highlight the importance of the components of this particular pathway for the production of GABA. We talked about how GABA is going to be removed by these transporters. GABA removal is also important for function. And so most of the GABA will eventually be converted to succinate, which will be metabolized further, again in the citric acid cycle for production of ATP. So there's a lot of tie here with the metabolic processes within the cells that you probably learned at some point in a cell biology or introductory biology course. And they are absolutely essential, these different metabolites, in order to produce these neurotransmitters. Just interesting information that your book provides for you. Even though GABA functions primarily as the inhibitory neurotransmitter in the brain, during early development, pretty interesting data in, I think, 2006 demonstrated that GABA receptors could actually, in immature neurons, so in the developing nervous system, could function as an excitatory response. And so the receptor itself would utilize the difference in concentration of chloride ions during development, where the inside of the cell would have high concentrations of chloride ions, and so they would be transported outward. And so you would result in a depolarization event versus an inhibitory event. And so, of course, that switches later as the synapse matures, and you can sort of document the change in chloride, or the change in the chloride current, whether it's an inward current or an outward current, and then also whether you have an excitatory response, so you can see here this excitatory postsynaptic potential, or whether you have an inhibitory one as the synapse matures. So I will leave you at that. This the slideshow is already a very long one. I know it's over 60 slides. So what's left are just some of the neurotransmitters that are less common, but they are interesting molecules that are utilized for neurotransmission. And so I encourage you to take a look at them, and then perhaps some of them you might be able to use for your neurological disorder final project if they are associated with different neurological disorders. And so what's left is just basically we're going to come back to some of these. So there are the endogenous opioids that we're going to talk about with pain. Somatostatin, that is, again, also utilized with pain. Substance P, which we often see in neurons that are important for digestion. Oxytocin and vasopressin, of course, being involved with lactation, childbirth, and pleasure or orgasm. Oxytocin, really interesting study that was just recently published, documenting the use of oxytocin in maternal care, and so behavior of basically mice that have given birth to babies having high spikes of oxytocin, obviously, during the birthing process. And so something about the high levels of oxytocin within the brain causing more maternal care behaviors to be present in the female mice that had baby pups, or had gone through a pregnancy and had pups. But then the females that had not had any pups, had no exposure to oxytocin, not showing any maternal care-like behavior. And so it was interesting because the researcher injected a large concentration of oxytocin into the mice that did not have children or did not have pups, and so saw a difference in the behaviors of those pups just simply by the injection of the concentration of oxytocin that you would see at childbirth or when the pups are born. And so some interesting neurotransmitters here. So we are going to come back to them, and we'll talk about them as we go through this semester. And I think that I will end there and encourage you to take a look at these last few neurotransmitters on your own. Let me know if you have any questions, and I will see you on Monday.